Welcome everyone, and thanks for joining our Arthur Rylah Institute seminar on navigating barriers, fish movement in modified rivers. I'm Sarah Garnick, Acting Chief Biodiversity Officer and ARI Director this week, and I'll be chairing the seminar with technical support from Andy and Jess. I would like to begin by acknowledging the traditional owners of the land I'm joining you from, the land of the Wurundjeri people, and pay my respects to the elders past and present. I'd like to extend that acknowledgement and respect to the traditional owners of the lands that we're all dialing in from today, and to any traditional owners and Aboriginal people who may be joining us online today. In today's seminar, we're joined by ARI scientists Dr Zeb Tonkin and Justin O'Connor to discuss how ARI aquatic research is delivering a greater understanding of fish movement in a highly modified river systems and providing water managers with the information they need to help optimise flows and barrier design. We'll start with the seminar from Zeb. Zeb manages the instream habitat and environmental flows program at ARI. Zeb has extensive experience in recruitment dynamics of freshwater fish, environmental flows, fish migratory behaviour, floodplain assemblages, threatened and pest species, and habitat restoration. His broad skill base includes collection and identification of adult and larval fish, external and internal tagging, including radio and acoustic telemetry, otolith preparation and interpretation, and data analysis. He manages numerous small and large projects involving a range of stakeholders. We'll have time for questions after each talk, so please post these in the chat. And thanks so much, Zeb. Over to you.
Thanks, Sarah. I'd firstly like to acknowledge a number of co-authors that you can see here and also at the end of the talk today, and also the Mallee Catchment Management Authority and Murray-Darling Basin Authority's Living Murray program for supporting this project. It's been underway for more than 10 years now. Both Justin and I presented these talks a couple of weeks ago at the Australian Society for Fish Biology, but we thought it'd be great to share it with a bit of a broader audience. So here we go. Firstly, I'd also like to acknowledge the traditional owners on the lands from where we're all meeting today, and also, and in particular, the first people of the Miller Mallee Aboriginal Corporation, where this work was undertaken. So let's jump straight into things. Movement of animals is a key requirement to complete key life history functions, such as spawning or for accessing foraging habitats, but it's also one of the key drivers or vital rates governing their population dynamics. And the knowledge of such is really crucial for management. This knowledge is super important when, as we usually are in the freshwater world, we're dealing with altered ecosystems where habitat loss and fragmentation can really compromise movement and potentially population dynamics. And unfortunately, knowledge on the movement of a species is usually pretty sparse, but throw in some really modified habitats that are often unique to a site or area, and we're often faced with some real challenges and shortfalls in the knowledge that we need to effectively manage a species or a site. So that leads us to this study where we've collected more than 15 years of movement data for Murray cod, one of those iconic species in the Lower Murray Lindsay Island Anabranch system, specifically to assess the environmental conditions associated with movement, the influence of a new regulating structure on this movement, understanding what movement can tell us about the impacts and recovery from a major disturbance event, in this case was a fish kill event, and how this information can fill really key knowledge gaps regarding broader population dynamics and inform flow and regulator management moving forward in the region. So let's look at our study area. So the Lower Murray Lindsay Island Anabranch system, it's a real diverse range of aquatic habitats and reaches in here. We've got the Murray main channel from Lock 6 to Lock 8 that's really modified in terms of it's largely characterised by big weir pools that essentially resemble really large lakes. And so under really low to average flow conditions, we've lost all our lotic or that flowing water habitat. And then we've got a few, as you can see in the maps, a number of anabranches like the Lindsay River, the Upper and Lower Lindsay River, Potawatomi Creek, that are quite flow stressed. They really only engage when the Murray River is flowing at high levels. But of most significance is the Mullaroo Creek that links in with the Lower Lindsay River to bypass Lock 7. It flows around the lock. as you can see in the image here. And back in 2015, the offtake of this creek, so this creek generally used to flow under, irrespective of what the Murray River did, due to a fixed crest, low-level weir. But back in 2015, this was replaced with a regulator and vertical slot Fishway that helps managers manage flows in the underbranch independent of what the Murray River levels were doing. So there's been a lot of work going on where they're trying to manipulate weir pool levels to get broader ecological benefits, but in doing so, that was affecting the creek. So by having this regulator on there, we were able to, or the managers were able to actually manage what was going on in the creek. So irrespective of these changes in structures, the Mullaroo Creek, in particular the Upper Mullaroo Creek, maintains a considerable gradient and therefore stream velocities along a pretty long, extensive spatial span. And it's also got really great structural woody habitat in there. So basically this creek resembles what the main river channel used to look like prior to regulation. And as such, it's long been recognised as a really important habitat and sustaining an important population of Murray cod. So this guy here, one of our long-lived iconic species in the basin of huge cultural, recreational and conservation significance. These guys spawn and reproduce annually. They need flowing water habitats. So basically what we've lost in the Murray River in most years. They have got really strong links to structural habitat and juveniles and even larval conspecifics of the species. They all occupy they all have these same habitat needs, which is basically what that Mullaroo Creek's been providing. But what was going to happen to this habitat after this regulator's gone in, and there's been some of these alterations to the flow regime in there. And that's what this study is basically trying to find out. So considering this earlier understanding, there's been a whole lot of great research that went on back through the Millennium Drought. And then basically our original understanding of how things were operating for Murray cod in this study site, in particular Mullaroo Creek, was largely adult fish were found right throughout the system, but juveniles and larval life stages were only found in particularly the Upper Mullaroo Creek, with those life stages basically missing from the Murray River. Basically, we also had some early movement studies showed adult fish would move from the Murray River into the Upper Mullaroo Creek to access that favourable spawning habitat. But that's basically where our understanding sort of fell. And like I said, all this information was conducted when the Murray River was flowing really under really low flow conditions throughout the Millennium Drought. So that sort of moved us in what we did. We wanted to extend this knowledge base, particularly with the construction of the new regulator and management of flows. We've gone and tagged more than 200 Murray cod across all those reaches in the Murray Main Channel, in the Mullaroo Creek, and where we could in some of those other underbranches. We used both radio telemetry tags that have got a mortality switch in them, and also acoustic tags that largely went into juvenile fish. And we also incorporated some of those early movement data sets into our broader analysis. Along with tagging the fish, the region was separated into 12 zones using directional antennas and acoustic receivers. So basically at any one point in time, we can work out what zone a fish was occupying. And then basically with all the data we collected from this acoustic and radio receiver array, we assessed the probability of a fish moving between and among Anabranch and Murray Channel zones, and related that to discharge and weir pool height, the time of year, the regulator status, and also we were able to test what some of our recommendations were coming out of the study. So let's look at the period that we collected our data. So we were actually fortunate enough to capture a great range of conditions. So that grey shaded area is some of the historical flow regime that I just kept in there so you could see what sort of conditions that were present in the creek through time. Everything to the right of that was the time that we've collected our data set. So we can see at the start of the data collection period, that's that Millennium Drought period. And then we sort of rolled into the period where the regulator was constructed in 2015. We've had some fish kill events associated with the 2015-16 flood. and hypoxic conditions. We had some really strong breeding and recruitment conditions. And then we had more recently that 22-23 huge flood event as well. So we've got some really great variation in our environmental data to really test what's going on in here. So after all that, what did the fish do? In short, they moved around a lot, with nearly 90% of fish moving outside of their tagging range. We detected fish using every one of the study reaches at some stage throughout the project, and that included those almost ephemeral forest anabranches. But when they used them, the detection and the reaches they used varied a lot across years. So this is just a summary of that. This is an occupancy plot of what reaches these fish were using at a few key different, basically a dry period, a wet period. You can see a bit of a shift going on in what reaches these fish preferred. And I'll get back to this plot and the results on this a bit later on. So if we have a look at movement within either the anabranch system or the Murray River main channel, this was influenced by time of year, probably most strongly, real peak in movement throughout that around that October period. So almost September to December, but really peaked in October. That's the key spawning period. That's when our larger fish are trying to access their favourite habitat. But also discharge in the anabranch. So particularly when flows exceeded 1,000 megalitres per day in the Mullaroo Creek, the probability of movement within the anabranch was really high. And also weir pool height in the Murray River. Again, the higher the weir pool height, the more likely a fish was going to be to move across reaches within the Murray River. If we look at movement across the habitat types, so movement from the anabranch to the Murray River, again, this was influenced by time of year, so that peak in October, but discharge in the anabranch was a key driver of this. So basically when flows got really high, fish actually wanted to leave the anabranch and get into the Murray River. And I'll touch on that a bit later as well. If we look at movement going back the other way, movement from the Murray River back into the anabranch, so those movements that were really characterised what was going on during the Millennium Drought that I showed in that earlier conceptual model. Time of year, a big one, but the regulator status had a really big influence on this. And basically, we've seen a really big reduction in the likelihood of movement of Murray cod or adult Murray cod from the Murray River back into the anabranch system. It's almost 17 times greater in the pre-regulated period than it is post. But what was interesting is that sort of as time ticked on and we could manipulate what was going on with the regulator, the regulators actually got a construction where you can actually lay the gates down flat when the flows are elevated. And when those gates were laid down flat, you've opened up the channel and it's almost twice as likely for fish to move from the Murray River back into the anabranch when those gates were laid flat. So there's some information coming out there for managers. And a really unfortunate event took place in 2015-16 flood event where we got a really significant hypoxic event where dissolved oxygen levels sort of was a real sudden crash and DO levels dropped to less than 1 milligram per litre, and a hypoxic event and subsequent fish kill event occurred. Leading up to this event, big floods, we had fish were moving around our study reach all over the shop. Then we saw this rapid decline in occupancy. So we got the team out there to do some manual tracking. As I said, those radio tags have got a mortality switch in them. And they were actually able to find a number of these fish. So it was nearly 40% of fish were confirmed to have died during the event. We're finding these fish washed up in the floodplain here. You can see the tags sticking out of them there. You know, some fox predation and things with the carcasses. But on a positive note, after what we've seen out there, it was thought that pretty much everything there was mortality of everything out there. But on a positive note, more than 30% were confirmed to have survived. And actually, most of those fish had migrated down the Murray River and out of our study area. And then subsequently, they started to return in the following year or two. What was interesting, there was no influence on fish size or mortality. We generally, when these events occur, we just see these really large fish have died, and it's thought that they're the most susceptible, when actually it didn't matter what size fish we had tagged out there. They were all just as susceptible to some of these mortality events as the larger ones. So what happened straight after the event though? So we had some complementary population survey data coming in, and it sort of confirmed what our telemetry data showed. So this huge reduction in abundance within the study area of all life stages. But the year later, we did get, as I mentioned, a couple of large adults return. And despite this only a small number of adult fish in the system, we've had unbelievable reproductive event, recruitment event. So this was the year following the mortality event, and you can see here, and this is what we call a cohort or recruitment event. These persisted through, and they had absolutely unbelievable growth rates. The fish were nearly 18 months old, and some fish were exceeding 40 centimetres in length. So amazing survival, amazing growth rates straight after the event. So a big question for us was, from a movement perspective, what would these juvenile fish do? So we got out there and put some acoustic tags, which are a much smaller tag, into almost 70 fish when they were age 1 and 2. And we wanted to know what was going to happen with these fish in terms of survival and where they wanted to hang out. Well, the answer for a few years was actually not much. We actually thought a lot of these juvenile fish had rejected their tags or they'd been subject to mortality. The bottom plot here is just what went on with the fish after it was initially tagged. We essentially didn't detect this fish that was tagged in the Mullaroo Creek for a couple of years. And then all of a sudden, once the fish hit about age 4, 3 plus to 4, all our juvenile fish started turning up on our receiver array. And what it showed was basically these fish hung out, and obviously they needed to survive and grow. And then when they got close to maturity, and it coincided with the high flow event, it was time to move. Most of the fish in the Anna Branch system wanted to actually leave. The fish in the Murray system were still moving within the Murray system, but didn't want to come back into the Anna Branch system initially. But that was some pretty neat. We'd never really looked at these cohorts before. But what it sort of did was really highlight that broadly the Mullaroo Creek and the juveniles that it produces is a really important source population in terms of recruitment for the broader region. So essentially we need to keep, when we do get a strong recruitment event, we essentially need to keep them alive and growing and everything like that for another 3 or 4 years before they're ready to move and redisperse and find their own home ranges. And so this is some really important information coming through for managers in there as well. So while we're on this cohort that I've just presented, some really neat info just landed literally last week that we've been sharing, that came across our desk as a result of data sharing that's been going on across the southern Murray-Darling Basin via other acoustic logger arrays and other agencies that we have collaborations with. We do this a lot for species like golden perch and silver perch that we know move huge distances. And of course, we all share data to look at these big movement events. But one of those tagged Murray cod, we actually tagged this guy as a 4-year-old. So just before it wanted to do its move back in early 2022. And come that big flood event in 2022 in September, this fish sort of took off and actually left our study area in September, which, we see fish leave our study area. That was fine. But if we zoom right out, this fish actually got detected at Mildura a month or 2 later. It didn't get picked up on a whole range of loggers between our study area and Mildura, so it's obviously been using some of the flood runners and the floodplain to move around. Then zoom forward a bit further, it was then detected in the Nema River, 670 kilometres away. This fish had gone all the way up the Murray River, up the Edward Walkall River, and then into the Nema River, which was probably, as far as I'm aware, one of the biggest migrations or movements for a Murray cod that we've detected, at least in our study. And this was a fish that was only 50 centimetres, so small by comparison for a lot of the Murray cod we tag. Since that detection in the past year or 2, flows have really declined in the southern Murray-Darling Basin. And so our marathon swimmer looks like he or she is getting itchy feet and wanting to head back towards home. And they've since been pigged coming back downstream into the Murray River, and it was last detected down near Belsa Island. So it's nearly a 900-kilometre journey that this fish has done, which is pretty incredible for a Murray cod. So all of this information has been used to refine management actions aimed at enhancing movement that ultimately are linked to recruitment, survival, and broader population outcomes. So essentially, all those model outputs that I've sort of been through have been used to refine some of the timing and magnitude of managed flows. So we aim to deliver at least 1,200 meg a day in the Mullaroo Creek system to maximise fish coming in or actually wanting to access the Murray River. We've also got a minimum base flow recommendation, basically to keep fish alive and grow. We know that we've got really good survival when flows are maintained above that level. And also the regulator settings. So when we deliver that higher spring pulse, we also, along with the flow recommendations, we've got the gate recommendations where the gates are laid flat during that elevated spring pulse period. We've actually had a look at modelled what's gone on since we implemented these recommendations, and basically movement of these fish is 4.8 times more likely when the spring pulse is delivered. And now we're starting to see an increased likelihood of downstream movement through the regulator as the gates are increasingly being laid flat. So it could be that fish are starting to learn how to navigate this structure once more. So if we integrate all this new knowledge and circle back to our original conceptual understanding, let's look at our dry year scenario and what looks to be going on. Essentially, we've got a pretty healthy size structure within the Anna branch, but not so much outside of the reach, with mostly adult fish in the Murray Channel. These adult fish want to move into the upper Mullaroo Creek during elevated spring flows, hence our flow recommendations. But that passage, fish passage past the regulator is restricted unless we actually open those gates up flat. And then we actually get a much higher likelihood of fish being able to access their preferred spawning habitat during low flow periods. If we look at average to wet years, which is some of this new information that we've gleaned over the journey, we see adult fish actually prefer the Murray River, and new recruits redistribute into the Murray River and other Anna branches. This is some really new stuff, which is cool. We also, via both our movement study and also some genetics work that Catherine Harrison's been leading, some of those fish from the Murray system have been found to move as far downstream as the Chowla system and breeding with individual fish down there. So we've got some connection in terms of their genetics as well. And then there's those hypoxic events that fish remain in the Anna branch network at a really high risk of mortality. But those that are able to move out, so again, the importance of connectivity, a much higher likelihood of survival, and then eventually do return. Once water quality and our habitat starts to come back. And then, of course, after they do return, there's this really strong potential for really strong recruitment and growth to enhance recovery pathways. So some key messages here. Movement is influenced by time of year, flows, and infrastructure management. Movement has a really strong influence on population dynamics, and knowledge of such has successfully informed flow and regulator management. So yeah, as I said, there's a whole bunch of people that have been involved in this work. And yeah, we look forward to getting more information on some of these new cohorts that are moving through the system. Thanks.
Thanks, Zeb. That was really interesting. While I'm giving people some time to think, I've got a couple of questions. The first one is, I don't know why, but I'm always really interested when we manage to capture these long distant movements of animals, animals moving much further than we kind of think they could. Do you know kind of what other long distance movements might have been recorded for the species?
Off the top of my head, so I'll go back to some of John Kewen's early work. There's definitely for the species, for Murray cod, it's our really large fish, a fish over a metre or over at least 80 centimetres. Generally, we'll see the odd fish do a couple of 100 kilometres. Some of Jared Lyon's work at Yarrawonga, fish would move and migrate. There was one fish, I think there was a media article on this fish 10 or 15 years ago called Casanova cod, would always leave Lake Mulwala and head 1 or 200 kilometres up the Ovens River and go past Wangaratta. We'd never see this guy again, and then they'd do the same thing every year. But they were generally pretty rare. Generally, it's tens of kilometres, but you get the odd fish that does move those couple of 100 kilometres. But in these modified ecosystems, it's much rarer because there's that many barriers and things. It's tough for these animals.
Thanks. Yeah, I just always find it super interesting when animals, do things that we're not expecting them to do. I've got a more serious question as well. This is obviously a lot of really hard work bringing this all together over quite a long period of time. And I'm interested to understand, could this work be used beyond this initial application for planning, flow and regulator management at the site? What else have you got in mind for this work?
Yeah, absolutely. So what you said, this was originally set up to look at the impacts or role of flow and regulator at a local scale at that site. But more broadly, it's really informed long-term planning and where other similar structures are being implemented across the basin. We'll look at even simple things. I say simple about the proportion of fish that have died during these hypoxic events. That's crucial information to inform things like population models and stuff. That they're the key tools that we use to look at like 50-year impacts or 50-year trends in dynamics and things on the climate change or whatever it might be. So some really crucial information here for long-term sort of outlooks. Awesome.
Thank you. In the interests of keeping to time, oh, yes, great. We've got one question that just came in from Bryce Halliday, thanking you for the talk. And the question is around how important is the Mullaroo Creek in the context of Murray cod in the Murray-Darling Basin? And are there lessons learned there that could be applied elsewhere to help populations?
Yeah, so the Lower Murray population, super important population. In terms, and as we've seen, particularly in the Darling and some of those areas, those populations, those areas have really been hit hard under, some of the localised droughts and things we've seen in recent times. So the Southern Basin, some of these populations are increasingly becoming crucial. I guess what this work also shows is that, probably more than a decade ago, it was, termed the Mullaroo population of Murray cod. What this work shows, it's more of a regional population, but the Mullaroo Creek provides vital habitat during low flow periods and things to keep that population ticking. So yes, it's super important, but its connectivity with the broader population is just as important. So yeah, that's the main way to view it, I guess.
Great. Thanks so much. In the interest of time, I'll hand over to Justin. So Justin leads the Aquatic Connectivity Program at ARI and is interested in a holistic approach to riverine management. His research areas include connectivity, environmental flows, productivity and habitat restoration, which are all intrinsically linked, and the influence of each of these on the successful restoration of riverine fish populations. And I'll hand over to you now, Justin. Thanks.
Thanks, Sarah. Look, today I'm just going to talk about a Fishway project, Fishway construction project that we're involved with in the Yarra River in Melbourne. I'll talk a little bit about the history of the site and the construction of the new Fishway, some monitoring we undertook that showed that the Fishway wasn't working to capacity. It flows, river flows of about 800 to 1,000 megs per day, and how we went about identifying the issue and then rectifying it. And then some follow-up monitoring that showed that the remediation worked, and then just a few of the lessons learned. Firstly, I'd just like to acknowledge the Wurundjeri people whose land this work was undertaken on, and respect to their elders past and present. And also acknowledgements to Melbourne Water, who are the funder of this project, and all the co-authors who are involved in the project. So some background. As I was saying, this is a weir that was built on the Yarra River over 100 years ago, and it was built to drive the Dietz flour mill. It's a major barrier to fish movement. The weir itself is about 3 metres in height, and so for fish moving up and down through that section of the river, it's a major problem, and particularly for the diadromous fish species, and they're fish species that need to move between the marine environment and freshwater environment to complete their life cycle. This is just a photograph of a map of where the weir is located. You can see from the inset down here, pretty close to the CBD of Melbourne, is Dights Falls, and all that area to the right-hand side is the catchment. So the weir itself blocks hundreds of kilometres of river and waterways. These are just some of those diadromous fish species that I spoke about. That includes the federally threatened Australian grayling. These diadromous fish species, they typically live in fresh water as adults, and then once a year they'll spawn, and the eggs and larvae go out to sea, and they remain there for about 6 months, and then they move back into fresh water, where they remain for the rest of their lives. So you can imagine a 3-metre high barrier located down at the downstream end of the catchment can be a major problem for these small juvenile fish, which are about 6 months of age and about 30 to 50 mil in length, moving back upstream. And the Dights Falls weir has basically has changed the distribution of those fish over the years upstream of the barrier. There's been a few attempts at putting fish passage on the structure, and they've always been rock ramp designs. And, for a 3-metre high weir, a rock ramp design is really at its limitations of what you can pick up with a rock ramp. So they had limited success and limited long-term success. And usually what would happen with these rock ramp fishways, you get a high flow and the rocks would be moved in that, and they would lose their functionality. So back in about 2012, I think it was, the original weir needed to be replaced. It was still the old original pylon structure. It needed to be replaced, and along with that, there was flagged for a fishway to also be built. So after lots of consultation, it was decided on a dual fishway to be constructed, and that included a rock ramp fishway, lead-up fishway, which picked up 1 metre of height, and then a vertical slot fishway which picked up the other 2 metres of height. This is just an aerial photograph of the finished fishway complex. You can see up here, that's the vertical slot fishway, and down here is the lead-up rock ramp fishway. It's got 4 main ridges in it, leading up to the vertical slot. This is just a photograph of the vertical slot once it was completed. Downstream, looking up from that top photograph, the main thing to take out of that is this notched weir here adjacent to the fishway entrances, and that was built to improve flow adjacent to those entrances and therefore attraction of fish to them. And then in the bottom weir, it's a bit more of a close-up, you've got your notched weir there, and these are the 2 entrances. There's one in the corner and one in the middle. The vertical slot fishway itself, it was really quite conservative hydraulics, and that's because these small fish, the swimming ability of a small fish isn't that great. So you need fairly conservative hydraulics for those fish to be able to navigate through there. So it had 25 pools of about 76 mil head loss and a slope of about 1 in 30. And then each of the pools between the slots were about 1,800 by 2.1 metres, so they're quite large, and that was all around about trying to keep turbulence reduced to help those small fish being able to navigate through there. So once the fish well was completed, we undertook some monitoring. Basically, that was all about demonstrating the structure is working. If it wasn't, we could tweak the existing design, and any learnings from all of this can go into improve future designs. Common glaxias were the indicator sort of proxy species we use, and basically that's because every spring there's literally millions of these fish, about 30 to 50 mil in length, moving back upstream from the marine environment into fresh water. And they're a good proxy because they're pretty much the same size and similar swimming ability to a lot of the other diadromous fish species also utilising the fishway. So our sampling design for the rock ramp, look, we trialled a few things to sample the rock ramp, but rock ramp fishways are difficult to sample quantitatively. Like I said, we trialled a few things, but we ended up settling on comparing the fish length at various sections of the rock ramp fishway. And the rationale behind that was that the smaller the fish, the poorer the swimming ability. So that if the fish at the exit of the rock ramp fishway, adjacent to the vertical slot fishway, were bigger than those down the bottom, it indicated the fishway wasn't working to specification. We also looked, so we're looking at the size of the fish, but also the diversity of fish that move through there. Now, as part of that, within each of those areas, and this is just an aerial photograph, that aerial photograph again showing these 4 areas where we sampled for common galaxids to take their lengths, and we sampled and we took 50 common galaxids from each of those sections on each occasion, and there were 9 sampling occasions, and we compared the lengths between the 3 to ensure the rock ramp fishway was working okay. For the vertical slot fishway design, we trapped the entrance and the exit. We were doing monthly trapping, and that was increased. I'm not going to present all of that now, but that was increased during the peak periods of migration, which is spring, when those small juvenile fish are moving back upstream. We undertook 1.5-hour replicates, randomised at the time of day. And once again, there was 9 sampling occasions undertaken there. This is just a photograph of the gantry and one of the cages that we used to sample the vertical slot fishway. And for some results, so this is the rock ramp fishway. There were 12 species of native fish collected and loads of fish, over 19,000. The vast majority were common galaxius. But importantly, the length range of those fish was from 12 millimetres up to about 800 millimetres for a short finned eel, and that met that original brief of passing small fish through the fishway. This is just some of the results of the rock ramp fish assessment. You can see there, they're the 4 areas where we would take 50 common galaxius from and compare their length, and there was no ecologically significant differences between the lengths of any of those fish species. And the stats back that up. For the vertical slot fishway, once again, we got loads of fish moving through. We got 12 native species, over 20,000 fish all up. Once again, the vast majority were those common galaxius. And we had a wide length range of fish from about 25 mil up to 1,100 mil. And once again, that met those original design objectives. This is just some photographs of the species we were collecting. You can see there's lamprey there, Macquarie perch. These are some of the common galaxius that were coming through. At times, like at the peak of the migration, it wasn't uncommon to get up to 1,000 of these fish in a single 1.5-hour replicate. It was great that we got loads of these juvenile Australian grayling moving through, which was really good to see. And just where I've got highlighted there, there's a couple of chupong sitting there. And interestingly, prior to the weir, the fishway going onto this weir, there'd only ever been 2 chupong ever collected above Dights Falls fishway or weir. And we got quite a few of these fish moving through there, so that was really good to see as well. So the vertical slot fishway, for the length analysis, there was no ecological differences in the length of fish between the entrances and the exit. Stats also indicated that. However, where we did find an issue is when we looked at catch per unit effort against flow. And you can see from that graph there that when we got to flows of around 800, river flows of around 800 to 1,000 megs per day, the catch per unit effort really dropped off. This is all about the vertical slot fishway, at the entrance of the vertical slot fishway. Now, at the same time, when we were sampling the rock ramp fishway, we knew at those flows there were still loads of fish getting right up to the exit of the rock ramp fishway. So it indicated there was something going on between the exit of the rock ramp fishway and the entrance of the vertical slot fishway. And then when you had a look at what was going on with the flows just visually, when you looked at flows of greater than 1,000, it was pretty obvious that there were issues. This is just some footage of about 1,500 to 2,000 megs, I think. A bit loud. But that's the middle entrance. We're moving into the corner entrance here. That's the notch fishway. You can see there's really high velocities, lots of turbulence, loads of upwellings, and for a small 30 mil fish, that's not a particularly fish-friendly sort of environment. So what do we do about it? So we assembled a diverse team that included engineers, fish biologists, and RM managers, and we investigated different ways of resolving this issue. Some of that included baffles, lots of different baffle configurations, but also included the removal of the notch weir, although we didn't go down that path because the notch weir created some really good traction flow conditions at low flows, so we didn't want to interfere with that. We also threw up the idea of a new entrance, and then we looked at all of these things using CFD modelling to see what did and what didn't work. So this is just the outputs of the CFD modelling of different configurations, their average velocity and turbulence. You can see the original Fishway. Velocity is not too bad, but turbulence is really high, which is pretty obvious when you see that footage at 1500 megs. The other one to note here is this number 4 option. It looks pretty good, but the CFD modelling indicated there was lots of recirculation eddies at the entrance of the Fishway, so that wasn't taken up. So ultimately we went with the third Fishway entrance. So this is just a photograph of the original vertical slot Fishway. You can see original corner entrance and middle entrance, and it was decided to build a third entrance in this location here, along with modifications to the rock ramp Fishway. And this is the finished product. So yeah, this is the low flow entrance and a high flow entrance. And you can see that there was modifications of the the rock ramp lead up Fishway to accommodate the new location of the third entrance. So following all of that, we undertook some more sampling up to about 6,600 megalitres per day. And this is the results of that. You can see that original graph that I showed you previously, and this is the new post-modification graph. And you can see that up at these flows, we're getting loads of fish moving through. And importantly, if you have a look at that top graph here, the little graph here, the proportion graph, almost 100% of all fish are using the new entrance at flows greater than about 1,500 meg. So it indicated that the modifications worked well and the Fishway was working to capacity. So some of the key messages, it's really important from the beginning to try and get it right. And part of that is around about assembling a diverse team and getting as many opinions on the table as you possibly can. It's really can be quite expensive, both in dollar terms and ecologically, if you don't get this right. I mean, I think it was about 5 or 6 years in between this Fishway getting repaired and working to capacity. I think you need to, use modelling to inform the design, particularly on these larger projects, these really expensive ones. The modelling makes up only a small proportion of the cost, and it just gives you so much more confidence around what you're doing and what you're doing will work. I think the project has reiterated the importance of monitoring over a range of flows. If we had gone in there on just those smaller flows, we would have walked away thinking the Fishway was fine, but it wasn't at those higher flows. So monitoring is really important as well. And then finally, I just wanted to finish on improved weir wall design. I mean, a really important tenet of Fishway design is that the Fishway entrance should be located at the upstream limit of migration. Now, most weir walls are built perpendicular to the flow, and the upstream limit of migration might be 20, 40, in the case of the Murray River, 150 metres wide. And that's problematic for fish to find those small entrances of a Fishway. So, I think if we want to get serious about fish attraction in big, wide rivers, we need to start thinking about things like building a weir wall diagonal to the flow. So the upstream limit of migration becomes maybe 10, 20 metres sort of area. And alternatively, you could use a V-shaped or a rounded-shaped weir wall and have a Fishway on either side of the river. But I think they're the sort of things that we need to take seriously have a look at if we want to get improved fish passage in big, large rivers. And that's it. Thank you.
Thank you, Justin. That was really, really interesting talk, thinking about how we can improve things for our native fish. There's a couple of questions have kind of come in to the chat. There's quite a bit of discussion about the purpose of the weir and what different options you considered in terms of remediation. And so it's, I guess the question for you is, you know, we're talking about Fishway construction. Where might weir removal fit into this discussion? And, you know, under what sort of circumstances would you recommend that versus other alternatives?
Yeah, look, it's a really good question. It's something I probably should have touched on, but basically, weir removal should always be the first option. I mean, fishways are great, but they're never 100% transparent, and so weir removal should always be the first option. But a lot of these fishways are still functioning. These weirs are still functioning, and they still have a function, so it's not always a possibility. In the case of Dights Falls, yeah, weir removal was considered, but this is a weir wall that increased the height of the river about 3 metres 100 years ago. And in those 100 years, there's been lots of infrastructure because there's a weir pool standing upstream, been lots of infrastructure built around that. There's also a lot of social activities like kayaking, canoeing, that sort of stuff that goes on in that weir pool. So whilst it was considered, it wasn't really a viable option. But yeah, I do reiterate, where possible, the weir should be removed rather than a fishway put in.
Great, thanks. And I guess picking up on that kind of theme, you know, what do you see as the biggest challenges to improving fish passage across the state? Noting that, you know, we've had this history of kind of, you know, river regulation and changes and things like that.
Oh, look, it's a broad question, and there's probably numerous potential answers to it. I think, and I touched on it in this talk, is that fish attraction. I think for fish passage within Fishway, fish passage, I think we've got a fairly good handle on the sort of hydraulics that are needed within the fishway to get fish to move through. I think we've got a pretty good idea about their swimming abilities and stuff like that for a lot of fish, not all of them. So I think that fish attraction, I think, is one, and it's a worldwide issue. Everywhere in the world, fish attraction is seen as a big challenge. You know, if we can get the fish to the entrance of the fishway, we've usually got a pretty good handle on how we can get them through it. But getting them to that entrance, particularly in big wide rivers, is certainly a challenge.
Thanks. There's a question in the chat asking about, do you have any understanding of native diadromous fish that don't make it back upstream as a result of the weir? Do we have any information about that?
Oh, look, not specifically, but they're probably predated upon. I mean, quite down at most weir, if you go to any weir wall in coastal Victoria during spring, during the migration season of these fish, you'll see plenty of birds around. So there's probably a lot of predation going on, and also fish predation as well. Some of them would survive, certainly, and some of them do survive in those lower reaches. But the carrying capacity of that area compared to the entire upstream catchment, I'm assuming, would be quite a bit different. So, yeah.
Thanks. Were there any more questions in the chat? For those who might possibly be unable to see the chat, there was a question around, has this work been written up, and Tess very helpfully popped in papers from Justin's talk, some links there, but also paper from Zeb's talk. If anyone's unable to access those, please let us know and we can help you out there. Were there any other questions before we wrap up for today? All right, well, thank you everyone for attending today. Our next seminar on the Platy Patch Platypus Recovery Initiative is scheduled for Monday, the 27th of October. If you'd like a reminder and the link to the seminars, this particular seminar's recording, please sign up to our ARI websites. And yes, you can find all our seminar recordings on our website for any talks that you might have missed, including this one for today. And we'd also really value your feedback or any suggestions through the ARI Seminar link that we've just posted in the chat. So thank you all for your attendance today, and thank you to our amazing presenters, Zeb and Justin, and our tech team of Andy and Tess as well. And thanks to our great audience for turning up and asking interesting questions. Thanks, everyone.